Okay, so we've talked about atoms. We've talked about how two atoms can join together either by an ionic bond or a covalent bond to form small molecules. We talked a little bit about water as a small molecule. And really, we're ready to talk about bigger molecules. So macromolecules or biomolecules, those really mean the same thing. And all biomolecules that we're going to talk about, all biomolecules are organic compounds. And the word organic means based on carbon. In other words, carbon is going to be the main structural backbone of every one of these molecules. So biomolecules are all organic compounds. And the other things that are important to life would be water and some of the other trace elements. We're going to talk a lot about the organic molecules, which are carbon-based molecules. So organic in this context does not mean grown without pesticides, which would be a different way of using the term organic. In this case, organic means carbon-based. So when you take organic chemistry, it's chemistry based on carbon molecules. So the simplest carbon-based molecules are called hydrocarbons. And as the name implies, the only atoms that are involved in a hydrocarbon molecule are hydrogen and carbon. And so we start with those as our basic biomolecules, thinking about organic molecules. And so these would be some examples of hydrocarbons. So the gray shape is the carbon, and the white, those are the hydrogens. So these are like what we would call ball and stick models of molecules. And so you can see on the left, the gray in the middle is the carbon, and then 4 hydrogens. And then each of these sticks is a covalent bond. That's what it represents. So methane, ethane, ethene. And we're not going to learn how to name these, so you don't need to worry about that. But can you see that they are all carbon and hydrogen molecules? And remember that carbon always wants to make 4 bonds. So you can double check, look at the carbon in the middle, for example, and see if there are actually 4 bonds sticking out of it somehow. And then each hydrogen only wants to make 1 bond. So you can double check these, and they all have the right number of bonds. Some hydrocarbons will make ring structures, so these sort of circular structures that connect back to itself. And so you can see here, there's a 5-carbon ring on the left, a 6-carbon ring over here, and then another 6-carbon ring. And these have different names, but you don't need to memorize the names. But these are all types of hydrocarbons because they're all made only of carbon and hydrogen. And each carbon makes 4 bonds. From each carbon, there should emit 4 bonds from it, and each hydrogen makes 1. All right. And then on the far right, you see an example where something else can be in the ring. So nitrogen, you'll remember, makes 3 bonds. And so it can be part of the ring. The other thing that can be part of the ring sometimes would be oxygen. It can be in the ring. But on the first 3 in this picture are strictly hydrocarbons. The last one is a hydrocarbon mostly, and then it also has a nitrogen. But hydrocarbon makes up most of the structure of a lot of the molecules that we're going to look at. All right. Now, we talked earlier about isotopes. Isotopes are atoms of the same element, but they have different numbers of neutrons, so they have different weights. Isomers is not referring to individual atoms. Isomers is a term referring to molecules. So we're already looking at a different level of the hierarchy of biology, because isotopes refers to comparison of atoms, whereas isomers is a comparison of different molecules, but molecules that have really the same composition, the same number of each type of atom, but just connected in different shapes. Okay, so that's what an isomer is. For us, the most interesting isomer actually is called the enantiomer, this very last one on the list. Enantiomer. Enantiomers are isomers, but specifically they are isomers that are mirror image molecules. And mirror image molecules, if you look at the picture on the right side here, you can see this isomer, it's called the L and the D. L is what we call the left-handed isomer and D is the right-handed isomer, but that doesn't matter. But what you notice is that if I drew a mirror in the center here, if there was a mirror right here, then could you see how these are mirror images of each other? So they contain the same kinds of atoms, but they're mirror image structures. It's interesting. The reason mirror image structures are important, I think, is when it comes to the way the body reacts to things. And mirror image structures is one example. Other kinds of isomers will have the same kinds of effects sometimes, but when you have molecules that have the same composition but not the same overall shape, they're not exactly the same, the body will react to them in different ways. So isomers, the body won't react to isomers exactly the same. It won't react to one isomer in the same way as the other. So it won't react to the L isomer the same way it would react to the D isomer. You would think that it's only reacting to the different types of atoms, but it's actually reacting, your body reacts to the shape of the molecule very strongly. And probably the worst disaster, the worst example of a mistake that was made regarding isomers is in the 60s, there was a drug called thalidomide that was created, or that was synthesized. And what they accidentally didn't realize was that one isomer of thalidomide was helpful to pregnant women, and it helped them sleep and such. So it was prescribed quite a bit. Usually the body just won't react to the other isomer at all. It's just completely harmless. But it turns out in this case, the other isomer of thalidomide, which would be mixed into any drug that was synthesized, had the effect of birth defects. So you can see in the picture the boy with the limbs that are malformed. And so the R isomer and the S isomer of thalidomide have completely different effects on the body. In the case of this story, the R thalidomide is the one they were going for in terms of getting the drug effect. But there's always going to be some of the other versions mixed into any synthesized drug, and they didn't know that it was going to cause birth defects. So it's one of the biggest drug disasters of history. Of course, it got recalled. But isomers, let me just go back. Isomers in general, that term refers to molecules that have the same chemical composition, the same number and types of atoms, but structurally they're different, and so the shapes of the molecules are different. All right. So I will ask you what's the difference between an isomer and an isotope. So, just make sure you're clear on that, because the words are very similar. All right. And thalidomide is a great example of isomers that both the two versions of the molecule had quite different effects on the human body. Now, the last thing in this chapter are what we call functional groups. Functional groups are small groups of atoms that tend to be attached as a group to hydrocarbons. So we talked a little bit about the hydrocarbons, the ones that make a ring and the ones that make a straight chain. And when you take a hydrocarbon and then different functional groups are covalently attached to that hydrocarbon, you basically have a building kit for any kind of biomolecule. And so we refer to these little attachment groups by names. And so you need to know the name of the functional group, the name of the group of atoms, the structure, and then the properties. So basically memorize this whole chart. Now, the letter R refers to the rest of the molecule, like the hydrocarbon that this functional group is attached to. So you don't need to memorize the R. The R doesn't mean anything. But an O is attached, you know, you have a molecule here in the red, and then it's attached to an O, which is attached to an H. That unit is called the hydroxyl group, and it is attached to a larger molecule. The methyl group is C with 3 hydrogens. The carbonyl group, that's how you pronounce it, carbonyl, is a carbon with a double bonded oxygen in between 2 other parts of the molecule. So R can be whatever, anything. Carboxyl is kind of similar to carbonyl, except it specifically has a C double bonded to an O, and then off of the C also an hydroxyl group. But we call the whole unit carboxyl. But functionally, it's really like a carbonyl combined with a hydroxyl. But when you see the whole unit, we refer to the whole unit as carboxyl. The amino group is an N, a nitrogen, and 2 hydrogens. The phosphate group is a phosphorus, P, that's a new one, double bonded to an O, and then 3 other single bonded O's, and then 2 H's. And sulfhydryl is the one that has the sulfur. All right, so the carboxyl group can be fully charged, and it acts as an acid. The phosphate group can be fully charged and acts as an acid. The amino group can be fully charged, but it acts as a base. So these 3 can be fully charged and act as an acid or base. Remember, an acid is something that releases hydrogens into a solution to raise the hydrogen ion concentration. A base is something that binds to free hydrogens in a solution, which removes those free hydrogens from the solution and therefore lowers the hydrogen concentration in the water. And that is the definition of base. The rest of these are polar, except for methyl, which is nonpolar. And the reason methyl is nonpolar is, remember, carbon to hydrogen, those are nonpolar bonds. So that makes the whole attachment group, the whole functional group, a nonpolar entity. All right. So we will practice filling in charts and rehearsing and practicing this, but ultimately you do need to know these. And that's the end of the lecture for chapter 2. There's a lot in here. It's a pretty dense chapter if you've never studied chemistry before. So a little bit of a crash course in chemistry, but not all the chapters will be this dense.